All right, welcome back to the deep dive. Today, we're diving into how life begins. That magical journey from a single fertilized egg all the way to a baby safe and sound nestled in the placenta.
Yeah, it's pretty wild.
It is, it is. And we got this source, development.pdf. Clearly, you folks are fascinated by this too. So let's break down these very first steps, really get into the nitty-gritty of what's happening in there.
Absolutely. And the very first thing that happens after that egg is fertilized is this incredible burst of rapid cell division called cleavage.
Cleavage. Okay. So we're starting with just one cell, the zygote. What does all this dividing actually do? How does this one little cell turn into, well, a person?
It's like imagine taking a lump of clay and rapidly chopping it up. You get smaller and smaller pieces, but they're all still clay, right? Same material. That's what happens with cleavage. The zygote divides and divides, but it's making identical copies.
So it's not like those cells are becoming specialized or anything yet?
Not at this stage, no.
Yeah.
This is all about quantity first. Eventually, you get this solid cluster of cells, all the same. And this is a stage called the morula.
The morula, right.
Ah.
I love that the source compares it to a blackberry. That's a pretty good visual.
Yeah, it is. And all this dividing keeps going until basically this little cluster, now called a blastocyst, bumps into the wall of the uterus.
The uterine wall. So what's the point of all these identical cells? The source contrasts this with how things work in salamanders, for example.
Ah, yes, that's a great point. In mammals, having all these identical cells early on gives us this amazing flexibility later. Each one has the potential to become any part of the embryo.
Wow. So we're setting the stage for some serious developmental fireworks down the line. And when the blastocyst meets the uterine lining, that's when cleavage wraps up.
Exactly.
Okay, so we've got this blastocyst. What does it actually look like? What's going on inside this little ball of cells?
Well, picture this. The morula, that solid ball, starts to hollow out. It becomes a sphere with a fluid-filled cavity in the center. This is the blastocyst.
Yeah.
Now, this sphere has an outer layer called the trophoblast, and tucked inside, there's a cluster of cells called the inner cell mass.
The trophoblast and the inner cell mass. Two names to remember. So what are they up to?
The trophoblast is like the gatekeeper. It separates everything inside from the outside world, and it's the first source of nutrients for the developing embryo.
Okay, so the trophoblast is like the early support crew, and the inner cell mass, that little cluster inside, it's protected by the trophoblast, but it's facing this hollow space, the blasticle. That's where things really get going, right? That's the starting point for, well, for everything.
That's it, exactly.
Exactly.
That little clump of cells, that's the foundation for the embryo. But first, that blastocyst has to find a home. It has to attach to the uterus. This process is called implantation.
Implantation. This tiny blastocyst latching onto the uterine wall, the endometrium. It sounds simple, but it's got to be huge for what comes next.
Oh, absolutely. It establishes that close connection that's needed for everything that follows, the formation of all the essential structures for the embryo to grow.
And the source mentions ectopic pregnancies, which is when implantation happens outside the uterus. A pretty stark reminder that this process has to be precise.
Absolutely. If the blastocyst implants in the wrong place, a viable pregnancy isn't possible, and it can even be dangerous for the mother. So, yeah, precision is key.
Okay, so implantation is successful. What happens next? The source talks about the amnion and amniotic fluid, which is what most people probably picture when they think of a baby in the womb.
Right. So after implantation, this membrane called the amnion starts to form around the developing embryo, and it creates a sac filled with fluid. That's the amniotic fluid, what we call the water that breaks before birth.
Oh, so that's what that is. So the amniotic fluid is more than just like cushioning, right? It must play a pretty important role in development.
It's vital. It provides a buoyant cradle for the embryo, lets it move freely and develop without restrictions. It's also a protective barrier, absorbing shocks and keeping the temperature stable. So, yeah, it's much more than just padding.
Makes sense. And finally, we get to placental circulation, the lifeline between the developing baby and the mother. This is where things get really intricate.
It is amazing. This is where those finger-like projections, called chorionic villi, come into play. They extend from the embryo side and make contact with the mother's tissues, intertwining with the uterine lining.
It's like they're reaching out, establishing a connection.
Yeah, exactly. And what's incredible is that these villi end up bathed in the mother's blood, which comes from tiny ruptured arteries in the uterine wall. This close contact allows for the exchange of everything the embryo needs: oxygen, nutrients, and removal of waste products.
Okay, so these villi are basically creating a huge surface area for exchange between the mother and the embryo. But one thing the source really emphasizes is that even though they're so close, The mother's blood and the embryo's blood never actually mix.
That's right. It's all done through diffusion. The good stuff passes from the mother's blood to the embryo's blood, and the waste products go the other way, all across a very thin barrier.
And incredibly, the mother and embryo can even have different blood types.
Exactly. And that blood travels to the placenta through the umbilical arteries and returns to the embryo through the umbilical vein. It's a completely separate circulatory system just for this exchange.
Wow, that's incredible. So, in a remarkably short time, we've gone from one single cell dividing into many identical cells, forming this specialized structure, the blastocyst, then implanting into the uterus, being enveloped by the amnion and fluid, and finally establishing this intricate connection with the mother's system through the placenta.
It really is mind-blowing when you lay it all out like that.
It is. And it makes you think, if two individuals with potentially different genetics and even blood types can interact so intimately through the placenta without their blood ever actually mixing, what other incredible biological boundaries exist out there? What other unseen, intricate systems are at work that we might not even fully appreciate?
That's a great question, and it really speaks to the complexity and wonder of life. It makes you wonder what other secrets are waiting to be discovered, what other incredible interfaces we might find as we continue to explore the world around us.
It really does. Well, on that note, we'll leave you to ponder that. Thanks for joining us on the Deep Dives. We'll catch you next time.
Thanks for having me.
Absolutely.